Mushroom research advances constantly, and the great discoveries may be around the corner. Unfortunately, life in a mushroom farm is always busy, and it's hard to stay up to date. Specialized scientific journals don't help due to technical language and frequent paywalls. Our mission is to bring the latest discoveries on mushroom science to the industry professionals and the mushroom people. Ladies and gentlemen, please welcome to the new episode of the Mycelium Digest.
Welcome back, fellow fungal enthusiasts, to episode 4 of the Mycelium Digest. We're kicking off something new today, a brand new segment we're calling our Featured Paper series. Basically, we're going to unearth a single, maybe groundbreaking piece of mushroom science, straight from the lab. Think of it like getting a backstage pass to the latest discoveries. We're pretty excited to share this with you. Oh, and if you're joining us on YouTube, please remember to like this episode and subscribe to the channel. And if you're tuned in on your favorite podcast platform, hitting that subscribe button and giving us a rating, well, it genuinely helps us grow this fungal community. So today we're unpacking a really fascinating study. It tackles a challenge I think, well, probably many of us have faced, right? How do you keep those beautiful, fresh button mushrooms looking and feeling great for longer? The paper we're highlighting is titled Glycine Betaine Treatment Extends the Shelf Life and Retards Cat Grounding of Button Mushrooms, authored by Parviz Maliksada and colleagues. And what really gives us confidence in this research, it was published in Scientific Reports. That's a highly reputable journal from Springer Nature, which, immediately signals this is high quality, rigorously reviewed stuff. So, what's our mission for this exploration today? We're going to break down how a relatively simple treatment could fundamentally change how we store one of the world's most popular edible fungi, could potentially reduce food waste, maybe improve access to fresh produce. Okay, let's get into it. You know the feeling, right? You bring home a lovely pack of fresh button mushrooms, maybe for a stir fry or something, pop them in the fridge, think they'll be fine. But then, just a few days later, that pristine white cap, it starts to discolor, maybe gets a bit fuzzy.
Yeah. Exactly. They just don't look quite as appetizing anymore, do they?
No.
And you're definitely not alone in that frustration. But on mushrooms, Agaricus bisperus, scientifically speaking, they're surprisingly delicate, especially when it comes to cold storage. They're particularly sensitive to temperatures below about 8 degrees Celsius, especially if they're kept for more than, say, 20 days. And this often leads to something scientists call chilling injury, or CI for short.
Chilling injury. Okay, that sounds kind of serious. What does chilling injury actually look like on a mushroom?
Well, the symptoms are pretty much exactly what you described. You'll see internal browning, maybe a somewhat fuzzy or even leathery texture on the outside. And sometimes the mushrooms just, they fail to develop their proper ripeness or firmness. It really diminishes their overall quality, appearance, texture, even the nutritional content suffers. It's a major hurdle for effective refrigeration and, getting a decent shelf life.
Okay, so the problem's clear. They're sensitive, they get damaged by the cold. What have researchers tried before to stop this? The browning and spoilage. What were the old methods?
Yeah, historically, various approaches have been looked at. Think about maintaining very specific low temperature conditions, which can be tricky, or even things like hot air treatments. Researchers have also messed around with chemical applications like methyl jasmonate or maybe oxalic acid. And while some of these methods showed, a degree of promise,
there was always room for improvement.
Exactly. There's always been a clear, pressing need for more efficient, maybe more natural techniques to control or at least diminish chilling injury in these white button mushrooms during cold storage.
And that right there, that's where this paper comes in. It introduces a compound called Glycine Betaine, or GB. So what exactly is GB and why is it relevant here for mushrooms?
Glycine betaine, yeah, it's a fascinating molecule. So it's called a quaternary ammonium compound. Just think of it as a specific type of organic molecule. Pretty stable, plays a big role in lots of biological systems. What's really cool is its natural function in plants. It acts as an osmotic regulator.
Osmotic regulator, meaning?
It means it helps plants protect their tissues from environmental stresses, like extreme cold or drought conditions. Basically, it helps them maintain the right water balance within their cells. It helps stabilize cell membrane structures, protect important proteins inside the cell from damage, and regulate the internal pressure. It just bolsters the plant's overall resilience, makes them tougher.
So it's like a natural shield, a kind of internal protector for cells. That's pretty neat. Has this been looked at for cold stress before, or is this a totally new idea for mushrooms?
No, it's not entirely new in the cold stress world. Applying GB externally, exogenous application, that's actually proven quite effective in reducing cold stress damage in various plants. Things like Arabidopsis, which scientists use a lot, and even in fruits like strawberries.
Ah,
And yeah, when it comes to mushrooms specifically, some prior research has suggested GB could improve their cold resistance. It seemed to stimulate the mushroom's energy levels and increase its own internal GB, along with other protective things like aminobutyric acid and proline. But the precise mechanisms, how GB specifically impacts things like the browning process or sugar metabolism in cold-stored mushrooms, that was still largely unexplored. So this paper really aimed to fill that important knowledge gap.
got it. So this research by Malexada and colleagues, they wanted to really pinpoint those specific cellular and molecular reasons why GB might help. How did they actually set up their experiment? What did they do?
Well, they were quite meticulous. They got commercially mature button mushrooms. Then they treated these mushrooms with different concentrations of glycine betaine. They used 0, 5, 10, and 15 millimole.
Right.
And the treatment itself was pretty simple. They just immersed the mushrooms in these different GV solutions for about 15 minutes at room temperature, 20 degrees C.
Okay, just a quick dip.
Yeah, then after a quick drying step, the mushrooms were packaged up and stored in the dark. They kept them at a consistent 4 degrees Celsius for 15 days. And throughout this storage period, they pulled out samples at 5, 10, and 15 days to, rigorously check various quality factors.
Okay. And what did they find? Let's start with the big one we all noticed first, the cap browning, the visual appeal. This is where it gets interesting, right?
Yeah, the findings here were quite compelling, actually. Sure, all the groups showed some visible color changes over the 15 days, that's expected, but it was the control mushrooms, the untreated ones, and even those treated with the lowest 5 mmGb concentration. They showed a really significant and increasing trend in browning.
Okay.
But crucially, the mushrooms in the 10 mm and 15 mmGb groups, they showed remarkably less color change, both outside and inside the mushroom. And the most impressive result, the least amount of browning overall, particularly right at the end on day 15, that was seen in the 10 mmGb treatment group.
Wow, okay, so 10 mmGb seems like the magic number, the sweet spot for keeping them looking good. But it's not just about looks, is it? What about that chilling injury itself and the overall texture, the quality?
You're absolutely right. Appearance is just one part. The 10 mmGb treatment also significantly reduced how much chilling injury occurred. To give you a concrete example, on day 15, the control group, the untreated ones, had about 24.6% chilling injury.
Okay, nearly a quarter of them.
Yeah, but the 10 mmGb group, only about 11.5%.
That's less than half. Huge difference.
It really is. Imagine cutting chilling injury almost in half just with this treatment. That's a potential game changer for shelf life. And beyond that, they also saw that the GB-treated mushrooms lost less weight over time, and maybe even more importantly, they stayed significantly firmer throughout the whole storage period. Again, 10 mmGb really stood out as the most effective.
So your mushrooms stay plump and firm, not sort of sad and spongy.
Exactly, which is what you want, right?
Absolutely. That's fantastic news. Great appearance, great texture maintained. But okay, beyond what we can see and feel, what was actually happening inside the cells, what's the chemistry that led to these improvements?
That's an excellent question. And it's where the study offers some really deep insights. The research found that 10 mmGb consistently and significantly reduced something called electrolyte leakage.
Electrolyte leakage.
Yeah.
Okay, what does that mean?
Think of a cell like a tiny balloon filled with water and important stuff, electrolytes. When the cell gets stressed or damaged, like by a cold, its outer membrane starts to leak, and those electrolytes seep out. So less leakage means the cell membranes are healthier, more intact.
Got it. Stronger cell walls.
Pretty much. By day 15, the control mushrooms had about 25.1% leakage. But the 10 mmGb treated ones, only about 14.2%.
Big difference again.
Huge. And similarly, they measured something called malondialdehyde, or MDA. MDA is a key marker for oxidative stress and something called lipid peroxidation. You can basically think of it as cellular rusting, damage to the fats in the membranes.
Okay, like rust. Not good.
Definitely not good. And the GB treatment effectively suppressed this MDA buildup. At day 15, control mushrooms had MDA levels around 14.3 units, let's say. The GB treated ones, only about 7.2.
Wow, cut in half again?
Yeah, it indicates significantly less oxidative damage to those critical cell membranes.
So GB is acting like a tiny repair crew, basically, keeping the cells strong, fighting off the cold stress. Makes sense for firmness.
Yeah.
But what about the browning specifically? What internal chemistry causes that?
Right, the browning. The researchers really dug into the enzymes involved there. Browning in mushrooms, it's mostly caused by an enzyme called polyphenol oxidase, or PPO.
PPO, okay.
What PPO does is it takes these naturally occurring phenolic compounds inside the mushroom. and it oxidizes them, turns them into other compounds called quinones. And those quinones very quickly turn into melanin, which is that brown pigment we see.
Ah, melanin, like in skin pigment.
Exactly. Same kind of molecule family. And the study found that 10 millimev GB significantly inhibited both the activity of this PPO enzyme and also the gene expression that produces the PPO enzyme in the first place.
So less PPO enzyme around, and the PPO that is there is less active.
Precisely. Less PPO overall means less browning. Simple as that.
Okay, that makes sense. But what about PAL? There was another enzyme mentioned, PAL. How did that fit in?
Yeah, PAL, phenylalanine ammonilyase. That's a really crucial counterpoint because, interestingly, applying GB actually led to an increase in the activity and gene expression of PAL.
An increase. Doesn't PAL make the compounds that PPO turns brown?
It does. PAL helps produce those phenolic compounds. But here's the twist, and it's key. When PAL activity increases like this, you get a higher buildup of these total phenolic compounds. And many of these phenolic compounds are actually powerful antioxidants themselves.
Ah!
So you combine that more natural antioxidants floating around with the decreased PPO activity we just talked about. You get this fantastic 1-2 punch that's really key to reducing the browning.
More defense, less attack.
You got it. The mushroom has more of its own protective antioxidants, and the enzyme causing the browning is less active.
Clever. Were there other protective compounds involved? Did GB boost anything else?
Absolutely. The GB treatment also effectively reduced the levels of hydrogen peroxide, H2O2. That's another common marker of oxidative stress inside cells.
Right.
So less of that is good.
Right.
And quite notably, it increased the content of other protective compounds like proline, which helps with stress tolerance, and ascorbic acid, which is just vitamin C.
Oh, wow. More vitamin C. That's a bonus.
Definitely.
Yeah.
And furthermore, applying the external GB also seemed to boost the mushroom's own internal or endogenous GB content. It kind of encouraged the mushroom to make more of its own glycine betaine. And it also increased the activity of an enzyme called betaine aldehyde hydrogenase, or BADH. That enzyme is involved in making GB.
So it's not just the GB you put on it, it actually triggers the mushroom's own defense systems to ramp up.
It certainly looks that way. It seems to prime its internal defenses.
Wow. Okay, so let's pull this all together. What does this mean for the mushrooms, for the farmers, and for us, the people buying them? It feels like GB is doing a lot of good things visually and internally, building up resistance.
Yeah, if we connect this to the bigger picture, glycine betaine is acting as what we call a crucial compatible osmolyte. That means it builds up inside the mushroom cells, helps regulate their osmotic potential, it basically manages their internal water balance. This allows the cells to maintain their turgor, their firmness, their volume, even when they're stressed by the cold.
Okay.
And this protective mechanism, it extends to safeguarding really important things like proteins and enzymes from denaturing, losing their shape and stopping working. And critically, it preserves the integrity of those cell membranes. The reduced electrolyte leakage and the lower MDA levels we saw, they directly support this idea of superior membrane protection and much less oxidative damage.
So it's not just a surface fix making them look prettier. It's fundamentally strengthening their core defenses against spoilage and breaking down. It's like deep internal reinforcement.
Precisely. And beyond that direct cellular protection, GB also seems to beneficially influence several key metabolic pathways. Now, while GB itself isn't maybe a direct antioxidant like vitamin C is,
Right.
its role in keeping the cellular structure stable indirectly cuts down on oxidative stress. It does that by minimizing the harmful overproduction of those reactive oxygen species, the things that cause damage.
Okay.
And as we discussed, by boosting that PAL activity and promoting the buildup of phenolic compounds, GB actively bolsters the mushroom's own natural antioxidant defenses, and that actively reduces the browning process.
So taken together, this GB treatment seems to offer a really powerful kind of multi-pronged approach. It extends shelf life, preserves the look, the feel, and maybe even the nutritional quality of button mushrooms during cold storage. It really does sound like a very promising strategy for post-harvest management. It can make a big difference in waste.
Indeed. I mean, this approach could significantly cut economic losses for farmers by just giving them a longer window to sell their produce. And it helps ensure that you, the consumer, get higher quality, fresher mushrooms that actually last longer once you get them home.
Yeah.
And what's also encouraging is that the benefits found here, they line up really well with similar positive effects seen when GB treatment was used on other delicate fruits. Things like bananas, strawberries, kiwis, even lotus slices, peaches, papayas.
Wow. So it's not just mushrooms?
No, it suggests a potentially broad applicability for this compound across a whole range of produce that suffers from similar storage issues.
That is fantastic. This one paper really gives us so much to think about. Okay, for our final thought then. If this research were to continue, what might be the next steps? Where did this go?
Well, building on this really solid foundation, future research could dive even deeper. You could explore the intricate molecular mechanisms using even more advanced tools like transcriptomics or proteomics.
Okay, looking at all the genes and proteins?
Exactly. To precisely identify all the affected genes and proteins, get the full picture of what's changing inside the mushroom at that molecular level. It would also be incredibly valuable, I think, to expand these studies to other economically important mushroom species. Does it work on creminous, portobellos, shoe tickets? And then there's optimizing it for real-world use, finding the absolute best GB concentration, the best application method for commercial scale, maybe even exploring integrating it with other existing post-harvest technologies, like, say, modified atmosphere packaging.
Yeah.
Could they work together?
Synergistic effects, yeah.
Potentially. And ultimately, of course, you need comprehensive sensory evaluations to people like the taste and texture of GB-treated mushrooms. And detailed nutritional analyses, too. Are we preserving or even enhancing the nutritional value?
Right, the full picture.
Absolutely. But it does make you wonder, doesn't it, if simple, naturally occurring compounds like glycine betaine could hold keys to solving some of these bigger global food waste challenges, maybe transforming things right from the farm to our tables.
It's a compelling thought. And that wraps up our first ever featured paper segment here on the Mycelium Digest. Today we delved into how glycine betaine treatment offers a really powerful way to keep your button mushrooms fresher, firmer, and definitely less brown during cold storage. We really hope this exploration gave you some valuable insights into the exciting and sometimes surprising world of mushroom science. If you found this discussion helpful or just intriguing, please do consider liking and subscribing over on YouTube. Or if you're listening on a podcast app, subscribing and maybe leaving us a rating would be fantastic. Your support truly helps us bring more of these analyses of fungal innovations to you. Join us next time for more fungal facts and fantastic findings. Until then, stay curious and keep exploring the amazing world of mycelium.